Okay, we're going to start our discussion of cells by taking a look at what's known as the cell theory. Now, the cell theory is a set of three statements developed in the mid-1800s, so around 1830, by several scientists, including Schleiden and Schwann. But basically, at this time, scientists didn't know the basic components of living things. And so there were scientists since about the 1700s that have been looking at things under microscopes. And so in 1830, they were finally able to say that, or postulate that all living things are made up of cells. So this includes all plants, animals, fungi, bacteria, etc. So everything on Earth that's alive is made up of a cell. That cells are the reason that things function, so it determines how they work and provides the structure of living organisms. So cells are the components, the building blocks of life. And this is the big surprise one, that all cells come only from pre-existing cells. So you have to have a founder cell in order to have any future cells. So this idea that cells come from pre-existing cells was a scientist named Redi. And so there were these interesting concepts in the 15 and 1600s about how things would originate. So one of the thoughts was that mice, pictured here, yes, these are mice, were created by a combination of dirty laundry and grains stored in your house, and that if you had these two things after 21 days, mice would spontaneously form. And so the preventative measure for mice was to not combine dirty laundry and grains in the same area. Now, we today would realize that you're providing habitat and food for mice, which would then come from outside. But what Redi did was he designed a set of experiments that had controlled variables, and basically his experiments, at least some of them are pictured here, and basically another sort of thought like the mice shown above was that flies would appear from any meat that was left out, and so that these flies would spontaneously be created by the process of the meat rotting. And so what Redi did is he set out two sets of jars of meat. One set of jars had no cover, and another set of jars had a screen or sponge cover. What Redi noted was that maggots appeared on the jars without the cover, and then flies would emerge from the jar, while the jars with no covers would not have any maggots and thus would not make any flies. And so the process of the flies appearing and the maggots appearing was happening because other flies were coming in and visiting the jars, depositing their eggs, and then their larvae grew, whereas the covered jars, the maggots couldn't get in, and so or the flies couldn't get in, so no maggots and then no future flies. So once we understand that we have the three tenets of the cell theory, those being that all living things are made of cells, cells are the basic units of structure and function, and cells come only from pre-existing cells, we then need to take a look at what all cells have in common. So we're going to break down some things about all cells. So what we have here is a list of some things that are common across all cell types. So all cells have that cell membrane. That's the barrier, the thing that prevents materials from entering and exiting the cell, the membrane. All cells are filled with cytoplasm, which is a special fluid that fills the cell. And so the cytoplasm over here is drawn in a sort of yellowish color, while the membranes are just the edges of these cells. All right, that all cells have some sort of genetic material. Okay, that doesn't mean they have a nucleus, right? But that they have genetic material stored in the nucleus of eukaryotic cells, so here, here, and here, that controls the activities of the cells and the cell's reproduction. And so in plant and animal cells, we can see these things with a regular compound light microscope. And so what we have here, we have an artist's rendering of cell and many of its organelles that we're going to go through in a later video. And then we have two what are called fluorescent light microscopy pictures of some cells, and so you can see some of the substructures, and these are really focused at the cytoskeleton, the structural components of these different cells in these pictures. So these are real pictures. These are not computer-generated images. So once we have an understanding of what all cells have in common, we're going to look at the first distinction or the first division of different cell types. That division is between what we call prokaryotes and eukaryotes. So prokaryotes are basically what we call bacteria. Okay, there are two types of prokaryotes. We'll get into that later. But bacteria cells are unique in that they don't have a nucleus. They have no membrane-bound organelles, and so they don't have things like Golgi and ER, okay, and other organelles that have or would be surrounded by a membrane. And we tend to view these as the oldest cell types. We have evidence from these types of cells going back as far as 3.5 billion years. And they're the smallest types. So with our compound scopes that we have here in class, you could just begin to see some of the larger types of bacteria. So good viewing for bacteria is in the 600 to 1,000 magnification range, which we don't have great access to here at our school. So continuing on with these bacterial types, they're fairly interesting. All the bacteria have a cell wall that surrounds them. That cell wall is often composed of this special molecule called peptidoglycan, which is a neat polymer of sugars and amino acids. That wall provides a lot of protection from harsh environment and other factors, as well as structure. Some bacteria have a flagella, this long tail-like structure that they can actually use to swim either in a water environment or through a slime that they secrete. Not all bacteria have a flagella, but some do. All bacteria do have that plasma membrane. That's the characteristic of all cell types. It is a phospholipid bilayer, just like in our other cells. And it does have some proteins embedded in it that plays a role in ions and nutrients and other material transport. Now, the thing to realize here is that even though these cells don't have membrane-bound organelles, they do still have things that carry out all these tasks. And it's basically, they do everything with enzymes and ribosomes. Okay, they store their DNA in what's called the nucleoid, which is just a region in the center of the cell, and then their cytoplasm is filled with all kinds of different enzymes and ribosomes that are taking care of the building and digestion of materials and all those other tasks that are needed for a cell. The other cell type then are our eukaryotes. Okay, eukaryotes basically means true cell. Okay, so the true life. These are the cell types that have membrane-bound organelles, and so they have a nucleus, right, that stores and surrounds genetic material. They have a lot of other organelles. So organelles basically means little organ or little organs. And so these are specialized structures that perform important functions. And so we just have some organelles listed here, things like ribosomes, the Golgi apparatus, mitochondria, centrioles, rough endoplasmic reticulum, smooth endoplasmic reticulum. These are things that have certain jobs to do in the cell. And so they have specific tasks, just like an organ system where they perform one function. We have evidence of our eukaryotic cells originating about 1.5 billion years ago. Okay, and these are the cells that make up protists, things like amoebas, plants, animals, and fungi. All right, all have this nucleus and other membrane-bound organelles. So as we take a look at these eukaryotic cells, the things that are really special about them is that they've divided up their tasks into these individual organelles. And so what these organelles do is they specialize in one function. So things like vacuoles store Mitochondria break down sugar into chemical energy. The nucleus houses the DNA. And so this division of labor among organelles equates to the division of labor among organs in your organ systems or in your body. And so that's why we get this word organelle, little organs, which is what they are for cells. Moving forward, what we're going to be doing is we're going to, in the next two presentations, go through all these different organelles, look at their task in the cell, what they do, how they function, and why that's important for each of the different cells.